We are now up to patient case study number 13. It's a female, 58 years old. She's been struggling with memory as well as some digestive issues. Go through, read the rest of the little scenario, create a patient profile for her on your data chart. As we look on the data chart, we need to go through color, clarity, as well as pH, and those are the first couple tests that we kind of want to do. And as we put patient number 13 into a glass tube, and we show you guys kind of that glass tube, this is what we're looking at. So I'm going to put it up against a white background just so you guys can see our color and clarity of what's going on here. So you're going to notice it's kind of a light solution. It's definitely got a little tinge of yellow to it, but not much. It's almost translucent. Clarity is definitely clear. I can definitely see through it. There's no cloudiness to it. Our next part of the data chart, you guys are doing pH. So we are doing two different pH scales, if you remember. So our first pH scale is the 0 to 14. Our second pH scale was a little bit more exact to it. So I'm going to start with the 0 to 14. I'm going to take that little strip. I'm going to dip it into our sample. And I'm going to match up that color that pH or the p hydrogen paper's turned. And I'm going to go, oh, it's probably about, I'm going to go 8.5 on that particular one in between those two. So now I want to get a little more specific. Remember, our second type only goes up to 8. So right away, I know that this had better turn a really dark blue or purplish color in order to make sure that that is correct. And when I dip that in and I look at my color, it's actually not even blue. It's actually more purple. So it's, I would say, higher than 8 on that particular scale. So maybe give it about 8.5. So it's good, both of them will match up. From there, we want to take our sample, our glass tube. We want to add our Benedict's solution to it. So if you remember, our Benedict solution is the one that's going to test for glucose. To our glass tube of 10 drops of urine, we are going to add 5 drops of Benedict solution, and we are going to go put that in the hot water bath. So that goes off to there for us. While we're waiting for it, we're going to take a look at the rest of our particular sample, and we're going to take a look at protein as well as chlorides. So across the top, you have Rollins's urine, and well number 1, just like what we did with our control patient the other day in the video and in class, first well is for protein. So we've taken 10 drops of urine, we added 5 drops of urette solution to it. In order for it to be positive, it would turn more a pink, purplish color to it. So we know Rollins, being that control patient, is negative for protein. Well, we did the same thing for test patient number 13. It also is going to be negative for protein. May look a little more blue to you, but however, if you remember, the sample itself was more translucent. So if I'm adding blue burette solution to it, it's not a chemical reaction. I need something that's pink or purple to change. It's more of a bluish solution because it's got more of that blue color to it because the urine was so translucent. So both my samples are negative for protein, both Rollins as well as patient number 13. In terms of my chloride, I added 1 drop of my chloride solution there with my 1 drop of urine, 1 drop of urine, 1 drop of chloride solution. They are both negative for that particular chloride as well. So far we have negative protein, we have negative chlorides, we have very high pH, we have translucent urine that is very light in color. So at this point we go back, we get our glucose tube off of the hot water bath, and we want to look at it. So we know Rollins is our control patient. That control patient is blue, kind of that turquoise blue. He is negative for glucose. So any change from that particular color is going to mean that we have a change in glucose. So on your lab, you guys will notice a little chart in the middle and says, okay, well, if it's turquoise, it's negative for glucose, no sugars pass through. If it's a slight green color, such as what you see with patient number 13, I'd give it probably about a 1+. Anything that's kind of an orangey green color, it's going to be 2+. Anything that's a dark brick red color is 3+ there. So not only want to mark plus, maybe kind of mark the color that you see there. So at this point, you guys want to go through, look at a microscopic view of that slide for patient number 13, see if there's any other information in there. Do we have white blood cells? Do we have crystals? Do we have red blood cells? What's going on with this particular patient? Bacteria, cells, whatever it may be. Copy and paste that, put that into your data chart, and then utilizing the data that you guys collected, analyze it, and come up with a solution or possible scenario of what this particular individual may have. You guys are going to follow your task material, and we'll talk about that a little bit later on.